So let's move on to our next presenters. Jennefer and Nikki introduced themselves earlier, but I'll tell you a little bit more about them. So Jennefer Kohler is a clinical and research genetic counselor at Stanford University Medical Center. She studied at the University of Washington and Johns Hopkins, and she's played an instrumental role in our diagnostic odyssey, really since we enrolled in the Undiagnosed Diseases Network back in 2016. And Dr. Legro is a second-year resident at Georgetown, as she said, and did a research internship with the UDN several years ago. And fortunately for us, showed an interest in our case and dug in and has stuck with it. And she was kind enough to come visit us in Seattle a bit ago. And it was that visit that really was the impetus for connecting us to our first other family, the Haddad. So we are really indebted to Nikki, and Jennefer and Nikki have pushed forward this publication that you're going to hear about now, and we're really, really grateful for it. So take it away, ladies.
Thank you, Jill, for the introduction. I think Nikki will drive our slides. So yeah, you're getting those set up.
I have like two screens up, so I'm like very confused.
It's always a juggle of the two screens and sharing your slides. But I think I can say as we get started, I'm thrilled to be here. I know Nikki is as well. And it's been really such a pleasure and a blessing to be able to work with Jill and her whole family all of these years. And just so impressive to see all the work that has been done and is continuing to be done. So I will get us started today, but Nikki and I were really hoping to focus on or share a bit more about the work we've been doing, which is more specific to what we're learning about the genetic and clinical features of people and families who have these loss of function variants in FAM177A1. So I'll start us off with some of the backstory and genetic side of things, and then Nikki will talk a little bit more about the clinical or medical features and findings. Go to the next slide. Thank you. Perfect. All right. I figured I'm a story kind of person, so I figured I would start us at the beginning of the story from our team's perspective. So this is a little visual representation of a family I think we all know very well, who was enrolled in the UDN study in 2016 because of a history of two siblings with global developmental delay, macrocephaly, autistic behaviors, hypotonia. Around the time of their enrollment, or around shortly after, both also had developed seizures. Next slide, please. So as part of the UDN study, like that, the research evaluation involves, a careful clinical evaluation, but it also does involve genetic testing and other research analyses that we think may be helpful in trying to identify a diagnosis. And I will say this, finding these FAM177A1 variants was not so straightforward. We had first sent clinical genome sequencing for the whole family. That was non-diagnostic. We reanalyzed the data on a research basis, meaning we were looking, I think, a little extra carefully. And that still was largely unrevealing. It wasn't until a second reanalysis performed with or through some of our collaborators at Hudson Alpha, looking specifically at larger chunks of the genetic data that could be extra or missing, that they identified these two kind of large chunks that were missing in both kiddos, and what we're going to call deletions in any case, and flagged that basically for us. And so we had something a little bit interesting to work from. Next slide, please. Okay, so this slide, I recognize that for many of us, this is going to look like kind of weird and very confusing. But what you are looking at is a visual representation of the genetic or genome sequencing data, and it's focused in on parts of the FAM177A1 gene as seen in our family. I'm not going to go into huge detail about what exactly you're seeing and how we interpret it, but I think that everyone can probably appreciate that you can see a lot of these little gray bars, and you can also see regions where there's a lot of white, which look like sort of areas where some of the gray bars are missing. And that's exactly what's going on here. And in this visual representation, what that says to us or is showing us is that there are deletions in these regions of the gene. Next slide, please. So at this point in time, once these two deletions were brought to our attention, of course, the next question is, okay, well, what does this gene do? Is it linked to disease? Short answer at that point in time was, we don't know much. It's not really been definitively associated with any sort of well understood health problem or disease. What we did know is that it's located on chromosome 14, as we just heard. The gene is evolutionarily conserved across species, which to us is a suggestion that it has some functional importance. We also know or could tell that it was expressed or turned on in many different tissues across the human body. I recognize that some of the words with these colorful bars underneath, these are all the different tissues that you can see the gene is expressed in. So I understand that the tissue names or labels are a little small, but what I want to point out is that on the left, the yellow bars are different regions of the brain. And so it certainly is also expressed in the brain. Next slide, please. So in terms of like gene function, what does this gene do? At the time, again, as I said, there really wasn't much known, and to date, there still isn't a ton known. There's a lot left to learn, but I figured I could give us an overview of what is known so far, and there's absolutely a lot of work ongoing to help us better understand the gene function. So there have been some what we call association studies that have linked the gene with certain health conditions and in like very specific disease settings. So this is not comprehensive, but for example, linked the gene to vascular inflammation in the setting of lupus and lupus-related cardiovascular disease, linked it to Graves' disease, juvenile idiopathic arthritis, and then a more recent study, I'll say the first three that I was just mentioning, they linked specific genetic changes within the gene to these particular health conditions. A more recent study was actually just looking at the levels of protein in blood and linked it to primary biliary cholangitis, which is an autoimmune disease of the liver. What these association studies, they start to give us a hint at the biological or health importance of the gene. But I do want to note that we have to be really careful about interpreting association studies because they don't necessarily indicate causality one way or the other. And as you can see here with these particular studies, they happen to be in very specific disease settings or with very specific health problems. And so the generalizability is a little bit limited too. More recently, and kind of interestingly, there are a couple papers that came out that were looking at the role of this gene in immune pathways. And both studies found that the gene acts as a negative regulator, of pro-inflammatory immune pathways. Essentially what that means is looking at these specific pathways in different ways. It was found that when there are higher levels of FAM177A1, that tends to dampen or lessen our body's signaling pathways that would create inflammation or an inflammatory response. So higher levels of FAM177A1 theoretically lead to lesser inflammation or inflammatory response. And whereas the opposite was also true, knocking out or shutting off the gene actually turned up or turned on those pro-inflammatory responses. Next slide, please. So while there's certainly some emerging information about the gene function, as I mentioned, there's still a lot left to learn, but we really were interested in these deletions that had been identified. And, despite not having a complete understanding of the gene's function, another way that we try to tackle these things is finding other people who have similar genetic variants and similar clinical features. And so through a case matching database, over a little bit of time, it wasn't immediate, we were able ultimately to match with multiple healthcare providers and families who had or cared for people with people with loss of function variants in both of their copies of the gene, and similar clinical histories, clinical features as those that we were seeing with our UDN family. Next slide, please. All right, so I was going to quickly run through what, I guess, the genetic piece of things before Nikki can take over and talk about the clinical side. What you're seeing here is I think I hacked this together, so forgive any errors, but a visual representation of the coding regions of the gene. And the numbered blue bars are what we call different exons in the gene. And then here with the orange bars, both vertical and horizontal, are the different genetic variants that we've become aware of or have been identified in people with FAM177A1 related disorder so far. As I've been talking with our UDN family, there were these two deletions, one on either side of the gene, so that where it says 8 kb del, 9.7 kb del, that's the representation of those deletions and where they correspond to in the gene. There is another family with a deletion of exon 3 in this representation. And then you'll see the horizontal orange bars, and the labels for those are, instead of larger deletions, they're either single letters that are switched out or a few letters that might be extra or missing. What is interesting to us about this, or what I think is most notable, is that all of these different genetic variants, the deletions and the smaller ones, are predicted to result in loss of function of the gene, either from cutting out large parts of the gene that prevent it from being made, or have it go through some cellular processes that are sort of like a quality check for our genes, or making the protein too short so that it can't function. But in any case, would be pretty confidently predicted to result in loss of function of the gene. And the reason I point that out really is because we don't always see such consistency about the different genetic variants that we're finding in a cohort of people. I think the last thing I wanted to point out here was what's shown here, I think I forgot to mention this at the beginning, so forgive me, is just one what we call transcript of the gene, or one version of the gene. So many of our genes will undergo different processing before they go on to make proteins, and that processing may result in different versions that may have different number of exons than others. And for FAM177A1, that is the case. And there are some versions of the gene that after processing have more than 5 exons. So I think it, as I have seen in the literature and on genetic test reports, it's a bit variable what different labs or different papers, et cetera, which transcripts they're using. So the specific numbers, exon numbers, variant numbers that you're seeing here may be a little bit different than what you're familiar with if you're a parent or a healthcare provider, or maybe what's been reported out, but they've all been kind of re-translated or interpreted to match this one transcript. So anyway, I don't want to clarify any confusion with that. I think that's all I had to say about this. All right, I will pass it to Nikki, I think, or did I have another slide? Nope, okay.
Okay. Yes, this is me. So my name is Nikki again, everyone we've met. So I'm going to be talking about the clinical features of FAM177A1 related neurodevelopmental disorder, which we're calling FAN, and open to suggestions. We've talked, Jen and Matt and I have talked a lot about this. And then, so I have a lot of clinical terms, which can be a little bit dry and simplistic and don't really capture what it's like to live with children affected by this disorder. So if any words need further clarification, please let me know. And then I want to try to bring this back to be patient and family centered, just so it doesn't seem like, you know, these children are defined by their clinical terms. Okay, oh, sorry. So this is our family cohort that we have in our manuscript, which was submitted. Jen and I had gone through a lot of trials and tribulations over the last 5 years of the identifying families and summarizing their clinical data. And based on international IRB regulations, this is the families that we are describing in our manuscript. And then these are pedigrees, which are representing the families and the affected individuals as annotated by the Black shading. And then I also want to say that for these pedigrees figures, you know, the stick figures cannot express what family life is like, what their journey to diagnosis was, which was years, I think, Jill, in your case, and then what the relationship is with these siblings who are unaffected. So the unshaded boxes here, you know, how rich their relationship is and how much they care about their siblings, and then the relationship between the siblings who are affected. And so that is that. And as you can see, this is a recessive disease, as the parents are unaffected, and then the children will have, the parents will have a 1/4 chance of having an affected child. I know, Jen, we had talked extensively about your genetic counseling and how they had told you it was a 1 in a million. And I think you told me that when Cooper was born, you said, I knew exactly that he was similarly affected. So that is that, but all of these children have siblings who are similar to them. Okay, so here is the nitty gritty, and then just broadly the characteristic clinical features so that all individuals share is global developmental delay, meaning that they have done things at a different age in life than what we would say typical development is. These individuals also have either have been diagnosed with autism or have autistic behaviors and behaviors related to that. So, you know, sometimes hyperactive, you know, very focused on certain topics and very sensitive to certain touch and feeling and things like that. In our cohort that we had just looked at, we have 4 females and 1 male, and they're all childhood to adolescent age, teenage now actually. And all of these individuals were noticed to have something different about them in infancy or childhood, which prompted further evaluation and diagnosis eventually of this disorder. And then, so we talked about the global developmental delay. Sorry, my notes here. And then these individuals, what they look like. So all of them have larger heads, macrocephaly, which is a very, almost every single person that we've or seen their records for have had this, and then characteristic facial features. So these children, they look like their parents. I just want to say that outright. They look most like their parents, but I've seen a few photos from the cohorts in general, and people have said that they have deep set eyes and then a prominent forehead. So they look like their parents, but also look like each other in some ways. And then 2 of our families, children have had seizures starting from childhood. And I want to say that These have been different types of seizures. They've been unpredictable, and they have been, I would say, Jill, I'm not sure what you would say, but very, very difficult to control and a huge stress. And then as far as other neurologic findings, we have spasticity, contractures, and hyperreflexia. So a little bit harder to control their limbs, and then some tremors. And then interestingly, some eye findings, congenital cataracts, which were corrected in early age, amblyopia, and then esotropia. So just a little bit different eye movement and control with that. Okay. And then we had, over the course of 5 years, collaborated with Dr. Reza Maroufian, and he had provided clinical data to us from 3 additional families, and he had given us permission to talk about these families today. They are not in our most recent manuscript due to the IRB and ethical regulations with sharing clinical data between institutions. All of these families, we're calling them D, E, and F, so just a continuation, have Middle Eastern descent. So families are from Egypt and Saudi Arabia, and then I think one is from Iran. And he had sent us some clinical findings. So just to go through them briefly, I've highlighted family F, who was the original cohort that was published in the Al-Azami et al., which had very few clinical details. But we had gotten more records from Reza, and I'll talk more about that. But just to briefly go through family D and E. So this double line here in their pedigree means that family D's parents were related in some ways. I believe they're first cousins. And we actually believe that family D, this individual, well, they did carry the homozygous FAM177A1 disorder. This individual passed away 20 minutes after he was born, and had multiple fetal anomalies on ultrasound, and likely probably had another disorder related to that pregnancy, in which clinical detail, this individual did not have a normal lifespan, and would probably be an outlier with the children that we know to have this disorder. And then family E, they have 2 siblings, also with a similar loss of function mutation. And I had just reviewed a few records from them, but very, very similar to the families we've talked about, and those cardinal features that we have, which is global developmental delay, macrocephaly, and autistic behaviors, so those big ones. And then lastly, family F, the parents were also consanguineous, I believe also first cousins, and they had 4 affected individuals. So 4 children, and all of them had very similar clinical features to what we had just talked about. And I think what was powerful about this data is that when Al-Azami et al. Had originally published a paper with first describing FAM177A1, and by describing, I mean 1 sentence, they had revealed a few clinical details, and then Dr. Maroufian had given us some more clinical data that I parsed through. And in the next slide, I think it's just easy to see this chart, which Jen and I had spent like painstaking hours going through every single record and like every single line of like what this could mean and contributing to the phenotype. And phenotype means just how they are and physical features and clinical findings. And so again, these siblings, they have big heads. They have done different things at different ages in their life. And then 2 of them has seizures. And then what we had really gotten from Reza was more resolution or more data on just what these behavioral differences are in them. So for example, these children were meant to have like low frustration tolerance, hyperactivity, sometimes irritable, sometimes aggressive, and sometimes emotional labile. But all of them were, I'm not sure if they had been formally diagnosed and evaluated for autism, but found to have like autistic features. All of them had trouble with language and learning that at a slower pace. And then 2 of them have had seizures, like we had known that our individuals have in this cohort. And then 1 of them had MRI abnormalities, and we don't know if that's just one that had gotten an MRI in general, versus the other 3 just not being evaluated. And in general, we know that the first individual, their firstborn, was evaluated extensively, whereas the other 3 had not gotten as much evaluation, which could give us like some slanted clinical details. But very, very similar. And then as far as the neurologic stuff, they had some tremors as well. And then issues with walking, and they're controlling all of their limbs. And then importantly, I just want to point out that 2 of them had recurrent respiratory tract infections. So like pneumonia at early age. It sounded like they're kind of like in and out of the hospital frequently as children, just being treated for pneumonia and other ear infections and stuff like that. And so with that, these are the families of the first one, which is Charlotte and Cooper, and then the additional family data that we had gotten from Reza. I think it was most easy to see in a table format that as we talked about, the clinical features are on the left, which most individuals are affected with. And then I'm just pointing out the recurrent infections. So a lot of these children have had respiratory infections growing up and ear infections. And I think if we had this disorder described and we knew that infections were a key part of it, many more would probably have had, you know, like, oh, we actually did have a lot of ear infections as a child. So that's how some bias could be introduced into this clinical data. And then, okay, so nothing more I want to say about that. And this is a concluding slide. We are open to suggestions of what everyone thinks, particularly the families, about how to name this and refer to this in the literature. We're going with FAND for now, which is kind of easy to say, and like FAM associated neurodevelopmental disorder. And we know this is what we've just talked about, that these individuals have look similar, they have similar behaviors, and then they have similar neurologic features. So those are the 3 things, and that's what we've described in our manuscript to really emphasize this. Most individuals have seizures, and this is a huge burden for the family and very, very difficult. And it's very, very hard to watch a child have a seizure, anyone have a seizure. It's heartbreaking. And I just want to describe that that is a big thing that these families have been going through. And then these families have recognizable facial features. They look like their parents, but they also look like each other. I haven't talked a lot about MRIs, but these, partially because I'm not a radiologist, and I know Elizabeth Haddad is here since she knows so much about radiology, but these, most people who have gotten an MRI, these families have white matter changes, and then 2 people have had delayed myelination. And I think based on what Jen had talked about for the scientific findings of that, we are seeing that FAM177A1 is a immune gene. So this, were we able to go back and reevaluate these children as they're growing up, we'd probably find a lot more immune findings. And I haven't touched on Cooper's autoimmune findings, but I think that if all of these children were to be evaluated again, that we would see a lot more immune findings. So that's what we're going to learn probably in the future for this. And then lastly, I just want to say that although this seems like a patchwork of different families, we know of probably, I think Jen, over the last 5 years, we've probably like 11 or 12 in total. But in terms of getting all of them together in one place and getting all their clinical data, that just comes back to the regulatory issues and the issues between collaborators. So with that, I will say, my last slide, thank you so much for having us here today. And this family has also brought Jen and I very close together. We've met like every other week for the last 5 years. So that's all I want to say, and I will pass off.
I'll just echo that. I want to thank everyone here, and honestly, it's really inspiring to see all the work from everyone, in the research space, in the therapeutic space, but truly with, I think, Nikki and our work, I couldn't be more grateful for the partnership of all of the families that we've been working with as well. Couldn't do it without you.
All right, thank you for that.
Time. There was one question. I know that we have a segue anyway to Jeff, and he had a question. question in the chat. So I'm wondering maybe we could address that maybe quickly before we jump to the next session.
Yes.
Jeff, do you want to just ask your question? I think it was a fair question.
Yeah, I noticed in a lot of the everything that was presented, you have a lot of siblings, and then the way it was explained on this call and also from our neurologist is there's a 1 in 4 chance of that your child having FAM, but so many siblings, and I was just curious. I think I kind of followed, but if you had any further kind of explanation on why you see that so much.
Yeah, so it is true that when two people who are what we call carriers for the same thing, and in this case it would be FAM, have children or have families, we expect that there's a 1 in 4 chance in each pregnancy that the child will inherit the 2 non-working copies of the FAM177A1 gene. So, in many families, just across disorders that work that way, what we call recessive genetic conditions, oftentimes there's really only one child in the family who happened to inherit both non-working copies of the gene and develop that condition. Why are we seeing in this cohort that there's a lot of siblings? I don't know, maybe there's a little bit of bias or what we would call ascertainment bias, where families who have multiple siblings affected by the same things may tend to get a more comprehensive genetic workup or present to care earlier, or essentially be identified faster than those where only one child may have that disorder. That's my hypothesis. But you're correct that it's a 1 in 4 chance.
I see.
I mean, sometimes you're going to flip a coin and there'll be a stretch where you'll get a bunch of heads in a row. So you have a few families here. So it's also the law of statistics with small numbers can kind of seem skewed. But I think the ascertainment bias is also a very, very, very powerful explanation. But appreciate you bringing that point up.
Cool. Thanks for the info.